Hello, everyone, and welcome to today's live broadcast, Clinical Implementation of Pharmacogenomics, Delivering Genomics to the Bedside, presented by Mark Dunnenberger, Director of Pharmacogenomics at NorthShore University HealthSystem. I'm Alexis Corrales of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your question into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Dunnenberger. I will now turn the presentation over to him.
Thank you for joining me this afternoon to discuss clinical implementation of pharmacogenomics, delivering genomics to the bedside. Throughout this presentation, we're going to discuss key elements to implementing pharmacogenomics and how we take this laboratory-based science and deliver it to improve patient care at the bedside. For disclosures, I've received honorariums from AmeriHealth. Please note, no material in this presentation addresses the services or products that AmeriHealth provides. Objectives: by the end of the presentation today, you should be able to define pharmacogenomics and related terms. You should be able to list gene-drug pairs ready for clinical implementation and describe components of a clinical pharmacogenomics program. In essence, you should be able to have a foundation to help develop a pharmacogenomics program of your own. I'll describe the health system that I work at, as I will use this as an example and a number of points that I'm going to make today. So NorthShore University HealthSystem is in the northern suburbs of Chicago, based in Evanston and going up basically to the Wisconsin border. It's probably a health system very similar to a number of people in the audience. It's a 4 community hospital system with about 800 beds. Importantly, we have a medical group that is over 100 offices, and these two, both inpatient and outpatient, are integrated together in the same medical record. We have about 40,000 admissions in a year, any given year, and a little over 120,000 ED visits in a year. Again, as I pointed out, we have an integrated electronic healthcare record, and we've had that for over 10 years now, so since about 2002, 2001. All right, so let's jump into some definitions. So pharmacogenomics, and I will interchange pharmacogenetics, and for the purpose of this presentation, you can think about those terms interchangeably. But it's basically how changes in your DNA affect the way you're going to respond to medications. When we think about what changes we're talking about changes in the area of pharmacokinetics, or what your body does to the drug. Think about drug metabolizing enzymes and drug transporters. These would be cytochrome P450 enzymes and different solute transporters. Additionally, we may talk about pharmacodynamic variation, which would be what the drug does to your body. You might think about this as immune responses with genes such as HLA-B, as well as drug targets such as OPRM1 and the mu-opioid receptor. Another key distinction to be made here is the difference between germline and somatic variation. So germline variation is the variation that you inherit from your mom and your dad. It's what you transmit to your offspring. These variants can be helpful in predicting pharmacokinetic and pharmacodynamic effects. Think about this as this is the stuff that we're testing. We can look at the DNA in your blood through your white blood cells via a blood sample or a cheek swab and know that that DNA is the same DNA that's present in your liver, where a number of these pharmacodynamic properties and pharmacogenetic properties occur with drug metabolism. This is in difference to somatic variation, and these are not inherited. These are ones we typically think about related to tumors, so it's variation collected over time. This is going to be really helpful in predicting pharmacodynamic effects of certain drugs. We think about tyrosine kinase inhibitors in different tumors gaining mutation that makes them not susceptible to different tyrosine kinase inhibitors. So for this presentation, we're mainly going to focus on germline variants and not somatic variants. Somatic variants is covered heavily in the oncology space. So this is thinking outside of that. So variation that you inherited from your mom and your dad, variation that if you test it when you were born and you test it when you die, if you use the same technique, you're going to get the same answer. And so that allows us to do some very interesting things, which we'll talk about. So what does pharmacogenomics look like in practice, just on an abstract level? So here we see a group of blue people, and this is basically representing patients of any given disease. These might be males who are between the age of 60 to 70, have a family history of coronary artery disease, and they're going in to see their cardiologist. The cardiologist recognizes that their LDL is high and their HDL is low. So they look about the same. We're going to prescribe them a statin. When we give them the statin, that group, what one solid block of patients actually turns out to be 3 or 4 different blocks of patients. We have the patients represented in green here. These are the ones that have little to no toxicity and get the benefit of the drugs. The ones where the drugs do what we expected them to do. We have the patients in gray here. These are the ones that get no therapeutic benefit from the medication. So if we gave them a statin, we would have them come back in and we would see that their cholesterol numbers did not change at all. We have patients in the red here. These are the patients that have adverse events and toxicities. This might be the patient in that follow-up visit who complains of muscle pain associated with their statin. These patients become a huge challenge because we have to treat both their toxicity and then potentially find an alternative effective therapy that can provide the healthcare benefits that we were looking for with the original prescribing. There's probably a fourth group, which I haven't included on this slide, which is the combination of the gray and the red. So the worst of both worlds, getting no benefit whatsoever and having toxicity. Needless to say, those last 3 groups, the drug we chose the first time was not the optimal drug for them. If we can do genetic testing before we give patients that medication, that blue group now begins to look a little bit different. We're able to identify patients who are likely at a chance of having a suboptimal response to those medications. So what do we do if we're able to identify those patients? Well, we have the green group, that same group as before. They're going to get that standard therapy. The medication we were thinking about giving them originally, the go-to medication that a prescriber thinks about every time they see a patient that meets those clinical criteria. For everybody else, we can choose an alternative therapy. And so that may be a dose adjustment or maybe choosing another medication. If I'm thinking about the statin drug class, I have 5 or 6 options that are commonly used today that we might choose between. And one of those may be the better agent for the patient depending on their genetic makeup. And so we're not talking about going from first line to last line therapy. We're talking about choosing amongst therapeutic equals. So this pharmacogenomics and pharmacogenetics and applying it to patient care today is not about creating new challenges for clinicians to tackle, but instead deliver new data for them to conquer those challenges they're already seeing in practice today. Practitioners today are already dealing with this genetic variation. They just don't know about it, and now we're giving them data to highlight and elucidate this might explain what they've seen for so long in their practice. If we do this appropriately, we're going to derive safer, more effective drug therapy. I think everybody can agree that's a great thing. We can also increase drug adherence. And one of the things that we know is black and white in medicine is if a patient doesn't take a medication, it's never going to be able to help them. So anything we can do to increase adherence should lead to better healthcare outcomes. It's a main role for a pharmacist to do a number of interventions to increase compliance. We know for a number of disease states like heart failure, if patients take their medications, we can decrease hospitalizations. Decreased hospitalizations will then lead to decreased healthcare costs across the system. So here we take the concept of precision, individualized medicine, and really begin to see the value of it on a population health level. And so this is a really interesting intersection of two major phenomenons going on in healthcare today: personalized medicine, precision medicine, and population health. And it really makes pharmacogenomics ripe for implementation today. So that's kind of the theoretical of how we would do it. Now, what are the steps that it actually takes to get it done? Well, to translate laboratory data to patient care, we have to go through a genotype mechanism. So this is looking at specific places in the DNA and actually getting those base pair changes. In the pharmacogenomics world, which is different than most of other genetics, we translate those into haplotypes recognized in the star allele nomenclature. So you see a star 1, star 2, star 3. This represents one half of what you inherit, whether it's from your mom or your dad. Then we put that together in a diplotype. This is where we combine what you get from your mom and your dad. So we describe a patient as star 1, star 1, star 1, star 2, star 2, star 2. And so I'm going to stop and pose a question to you. So who in the audience knows what CYP2D6 star 5 star 5 means? Many of you guys are going to have no idea what that actually means. And that's really the challenge that we have, right? Most clinicians don't understand star allele nomenclature. So we have to translate that one step further to a phenotype. And if I begin to tell you that a patient who is a CYP2D6 star 5 star 5, it has the phenotype of a CYP2D6 poor metabolizer, now you're beginning to get some meaning with that, right? We can begin to understand, oh, that patient's CYP2D6 must not function as well as the average patient. So phenotype has some clinical meaningfulness behind it, but we really get our bang for our buck when we get to the therapeutic recommendation, where we take that idea of a CYP2D6 poor metabolizer and say, hey, if you're going to prescribe codeine to this patient, maybe you shouldn't, because they're not going to be able to activate it in the same way as the average patient, and therefore not get the therapeutic benefit or the pain-relieving benefits of codeine. So this is the challenge that we're faced with in pharmacogenomics. You've got to walk through all of these steps and give people the data they need at the moment that they need it. And you can imagine each of these steps may be important at different points in the patient care arena, but most clinicians really care about that therapeutic recommendation. So you've got to deliver it in a real meaningful way. We take that on another level. What you see here is all the information you need to have to make a decision about somebody's CYP2D6 status and the drugs that are going to play a role. So again, I'll pause and say, who here can understand what this means and would know what to do in a patient care situation immediately just from this screenshot? That's right, a number of you, again, not quite sure what to do, and you can imagine if I gave this to the average ED physician, they're probably going to have no idea what's going on and get quite frustrated. So we do a simple translation. We turn this into a couple of sentences, and based off this patient's results, they're likely to have therapeutic failure from Paxil, and therefore we should consider alternative therapies for depression. Again, we get this idea of choosing between therapeutic equals. For treating somebody for depression, we have a number of agents we can choose between. So let's avoid the genetic problem if we can and go to another therapeutic equal. And that's what we're suggesting here with this alert. All right, and to really drive this point home, here's a patient case to show you where we could have used pharmacogenomics and how we could have used it. So this is a 13-year-old female that I saw in my clinic. She came in with her mom. The child was diagnosed with depression and anxiety, and her mom report numerous medication problems. Either they're not working or causing side effects. The mom talks about how every night she has to give her daughter poison before she goes to bed. And what is that poison she's referring to? It's the medications they're using to treat depression. So obviously they're not working, and it's creating a lot of social tension between the mom and the daughter. If we look at the patient's medication history, you can see here she's tried a number of common medications. So we have citalopram, sertraline, fluoxetine, aripiprazole, and venlafaxine. In the middle column, this is how it helped with her depression. So the red X represents no benefit whatsoever. The yellow X is maybe there was some benefit, but not completely resolved. And then over on the side effect side, the green check mark representing no side effects. And the red X is representing intolerable side effects. So you can see that for all 5 of these medications that they've tried over the last 18 months, they haven't quite worked, haven't gotten green checks across the board. So this is the challenge that I faced in my clinic. So in our pharmacogenomics clinic at NorthShore, we do a multi-gene panel. And the reason we do this multi-gene panel is we can test this patient once and get a number of answers that may be helpful in that patient's life throughout their care. Remember, for germline genetics, the pharmacogenomics that we're talking about here, if we do the test once and then we repeat it later on and we do the exact same test, we're going to get the exact same answer because this DNA isn't changing over time. So we have a multi-gene panel. For any given clinician, we're going to have to highlight which ones are important. So in this particular case, there are 3 of the genes that are important for this patient. So CYP2D6, CYP2C19, these have to do with the breakdown of SSRIs and some other antidepressants. And then SLC6A4, this is that presynaptic serotonin transporter, which may be helpful. So you can see it's represented by the diplotype. I told you I'd translate those into phenotypes. Here are the phenotypic translations off of these results. So a CYP2D6 poor metabolizer, a 2C19 intermediate metabolizer, and an SLC6A4 poor responder. So those are going to be 4 problematic phenotypes. And when we look at this patient's medications that they've tried, what her genetic results told us, and the description. So the key here is we didn't have this genetic information ahead of time. Because if we had, we would have known every medication we had tried so far wasn't quite right for her. So for S-citalopram, there's some genetic challenges with it, mostly related to the SLC6A4. We have ineffective therapy and may cause side effects. For sertraline and fluvoxamine, we see, again, ineffective therapy, more likely to cause side effects related to both how the drug is broken down and that SLC6A4. Aripiprazole and venlafaxine, likely to cause side effects because she's not going to break it down quite as well because of her 2D6 status. So at the end of the day, what were our recommendations? Well, consider SNRIs, so selective norepinephrine reuptake inhibitors. We gave some suggestions such as drugs as desvenlafaxine, duloxetine, and bupropion. One important highlight here is pharmacogenomics rarely is a crystal ball and normally doesn't lead to a single choice, but again, leads the clinician to narrow down their therapeutic alternatives and allow them to choose which medication might be appropriate. So to conclude with this patient case, with our recommendations, the clinician immediately went to try bupropion and risperidone, but it failed. And so that took him a month to figure out it was going to fail. And then we went on to the next recommendation. And this patient is now stable on duloxetine and lorazepam. Anybody want to guess why this patient tried bupropion first? This was the clinician's decision. Yep, I can see it here. Somebody got it right. So it was because of the cost of bupropion versus duloxetine. So it's going to be really important to think about when we're faced with a patient, it's about a patient care issue. So it's not just what do the genetics say, but what does the whole clinical picture say for pharmacogenomics? And that's going to be really, really important that we take the genetics and integrate it with the clinical answer so that at the end of the day, we're not left with 2 different answers, a genetic answer and a clinical answer, but we have one answer, and that's going to be optimal patient care. It's the combination of clinical data and genomic data integrated into it to make a robust recommendation for a patient. All right, so I think this is the question everybody wants to know. Who do we test for pharmacogenomics and when do we do it? So when we think about pharmacogenomics, for it to matter, you have to have a collision of 2 different patient populations. You have to have patients who receive high-risk medications. So these are medications that have a link to pharmacogenomics. So these are the ones where if I know something about your genetics, I can make a recommendation. And then you have to have a patient population where the genotype is actually there. So the problematic genotypes are present. So when we get an overlap of those 2 populations, then pharmacogenomics can actually make an impact into patient care. So let's look and see what size those two different populations are. So an audience response question here. What percent of the population has a high-risk pharmacogenomic variant? So we have 5%, 20%, 77%, and 99%. So I'll give a minute for people to think about this. And so, again, the question is, what percent of the population carries at least one variant in the pharmacogenetic genes related to pharmacogenomics? Is it 5, 20, 77, or 99%? So for those of you who guessed 99%, you got that correct. So this means almost everybody in the population carries at least one genetic variant. This data was calculated off of population frequencies. And this is data that's actually from my clinic. We can see in the clinic, in the real world, we have a 98% of the population carrying at least one clinically actionable variant. And what's really, really exciting here is most of the population has 2, 3, or 4 variants that are clinically actionable. And so what that means is by doing a panel test, we just don't get one answer, and once we have that one answer, there's really nothing else there. But we have multiple opportunities to make a difference in a patient's lives because there are genetic variants in multiple genes. And this supports doing a multi-gene approach as opposed to a single gene approach. So one idea is that population in the blue, it's huge. Almost everybody has a potentially clinically significant pharmacogenomic variant. So now let's look and see who's on high-risk medications. So in NorthShore, it's about 1 million drug orders in any given year. In the United States, it's about 750 prescriptions actually dispensed in 2013. So quite a lot. If we think about every patient has some genetic variant, then that means each of these prescriptions may have had a chance for pharmacogenomic data to make a difference. This doesn't say that all 750 million prescriptions corresponded to patients with the same pharmacogenomic variants. It's not that all of these could have made a difference, but they had the potential to. And there's a group called CPIC, or the Clinical Pharmacogenomics Implementation Consortium, that writes guidelines to help clinicians understand how to use genomic data if we have it. And so this group is a group of experts from across the world, and we work to write those guidelines, not answering the question of when should you test it, but instead answering how should you use genomic data if you have it. And the idea there is we tell you how to use it, clinicians should be able to best figure out how to implement it into their clinical workflows. So if we look at common drug gene pairs where CPIC has seen that the evidence is high enough to implement into patient care, we can see that there are drug gene pairs that span patient care all across the place. So we have TPMT and thiopurines. In an adult population, you might see this in rheumatological disorders. In a pediatric population, you might see this in acute lymphocytic leukemia. So cancer to rheumatology, clopidogrel and 2C19, that's going to be in the cardiology world. You'll see a number of things here related to psychiatry with phenytoin, with the SSRIs. Probably one of the most common medications prescribed in my institution on ondansetron is in the upper right-hand corner. You can see that the drug gene pairs are complemented by the red and gray guys again. If you remember from earlier in the presentation, red is likely to have adverse events, gray is likely to have ineffective therapy, and when both are present, then we find out both ends of the spectrum. So what we know about the genomics changes a little bit depending on the drug gene pair. I'll give you guys a minute to review this slide and pick out your favorite drug gene pair to do a little bit more research on in the future. Okay. So we've talked about a number of drugs that are going to be really, really important. We've talked about that most patients have pharmacogenomics. So in theory, you could test everyone, but then the question becomes, when do we test them? Do we take a preemptive approach or a reactive approach? And so to give you some descriptions about what we mean, this is not a one or the other. They're actually on a spectrum, and you can go from preemptive all the way on the left, and gradually go all the way to the right and get reactive. So the most preemptive approach that you could have is going to be a healthy patient who's on no medications, testing them with a multi-gene panel because they're interested in care. And so for this, the value of this particular test is going to be in the future. And every therapeutic decision where pharmacogenomics may play a role in the future, this type of testing has the most potential value in this clinical setting, and looking at it in these terms. If we move a little bit more to reactive, we have a patient who is starting an antidepressant therapy, and we're going to test them for a multi-gene panel. So for that antidepressant therapy, this panel is reactive, right? We're waiting till we need a medication, and then we're going to test them. But for everything else in that multi-gene panel, it becomes preemptive. We have that data available for this patient's care at some point in the future. Moving a little bit more reactive, we have a patient getting ready to start a Bacavir. We're going to test them for a single genetic variant, HLA-B5701. As we move over a little bit further to the most reactive, we have a patient who's failed every known therapy for a disease, and they're being tested to help explain why that happened. And so as we think about the value of this, well, it's not how we're going to use this pharmacogenomic data to make a new intervention, right? We've tried every therapy that's out there. Pharmacogenomics isn't going to tell us something new, but what it might do is add personal value to this patient. It may be able to explain to them why they failed some of those previous therapies so that they understand what is better going on in their care. So you can see on these spectrums of preemptive on one side versus reactive on the other, they both can be valuable to a patient, but the ways in which we measure the value will have to be different. It's going to be really important as you set up a program that if you are looking at clinical endpoints or the different outcomes you're going to look at, that your testing approach matches where you're going to find value. It's going to be incredibly important. So we take that preemptive look where we have the most potential clinical value because we know that patients, regardless of their conditions, may receive high-risk pharmacogenomic drugs from the previous slide. And we know that patients are likely to have at least one genetic variant from another previous slide. Let's go to the next level and say, okay, can we predict what % of patients are going to receive a high-risk medication for the first time over any given 4-year period? And so this is a study that was published a few years ago, and they looked at prescription claims data. And you can see represented over here in the red that if you're of Medicare age, there's a 50% chance that you're going to receive a new prescription for a high-risk pharmacogenomic medication over any 4-year time period. So that's incredible, a coin flip chance of getting a high-risk medication. So this means that most of your patients are going to benefit from pharmacogenomics if you do testing, if they're over a certain age, so 40 and above. So now we're beginning to hone in on what patient population we should do this type of preemptive testing. There are a couple of key points about this. So one is it's a high-risk medication for the first time. Your patient being on a high-risk medication may not be a reason to do pharmacogenomic testing. In fact, if your patient's been on a medication for more than 6 months, I probably don't care about their genetics because they've already told me what their phenotype is. They're already telling me whether that medication is working, whether they're having side effects, those types of things. And so genetics doesn't really impact that patient care delivery in that setting. However, if we get it ahead of time, before they have exposure to medication, we can then make a change. And then over a 4-year time period, that's going to be really important because that gives you an idea of how long do we have to measure the outcomes to see the benefit for a particular patient. If you have an implementation where you're only going to measure outcomes for a year, well, it's going to get a little tough to make sure that a large percentage of your population sees a high-risk medication. But if you do a 4 to 5 year study, well, now we're getting pretty good numbers into seeing the benefits of pharmacogenomics. So as you're evaluating the impact of your program, it's important to keep these numbers in hand. All right, so now I'm going to discuss what this looks like in practice and how we've done this at NorthShore. So some key components for a pharmacogenomics program are represented in this diagram. So that big blue oval, that is the pharmacogenomics program. And we've got 5 different components. So we have to come up with genetic testing. We have to collect that information, and that's the genetic database. We have to link that to some kind of knowledge base or clinical decision support tool, which is represented here as CDS. And then you have to deliver that to the end user, whether the end user is patients or providers. You can see that here as a chart. And at the end of the day, education becomes incredibly important as we do pharmacogenomics. So let's hone in on genetic testing and how we get that done at NorthShore. So at NorthShore, we have a dedicated pharmacogenomics clinic. This clinic was first started in March of 2015. And we started it because our patients wanted to use pharmacogenomics in their care, even though they weren't sure what that meant. And our providers wanted to use it in their patient care, even though they didn't know what were the best ways to do that. And so we set this we set this clinic up over 2 different visits. And so the first visit was really important for us to highlight the benefits, limitations, risk, and cost of testing for a given patient. And so this way, they know what they're not going to get, they know what they're going to get, they know how much it's going to cost them, and they can make a value-based decision on whether pharmacogenomics is right in their care. It's really important to have realistic expectations about what pharmacogenomics is going to do. As I said earlier, this is not a crystal ball. It's not going to explain every response a patient had to a medication. It's not going to give us that one answer that's going to solve all of their problems, but is a piece of clinical data that can be integrated into the rest of their care. So really important to have those realistic expectations. There's a lot of misinformation out there. We found this visit to be really, really important. In fact, it almost took an hour to begin with to get this going. Once patients decided to go forward with testing, we would test them in that first visit, and we would have them come back for a second visit to fully explain their results and really answer any questions they had about the clinical meaningfulness of their pharmacogenomic results. And patients found this one to be very, very rewarding. At the end of this, we were able to take these results and then share them with other providers based on patients being willing to share this data with their other providers. You can imagine that this approach isn't very scalable, right? I can only see so many patients in my clinic every day. And if pharmacogenomics should be beneficial to every patient, we've got to figure out better ways of testing them than having them come to the clinic and getting this all out of the way there. So what we did is we created this at-home testing kit. I mean, it's similar to a number of other third-party vendors where it's a cheek swab, or a saliva sample, depending on which test you use. And in our case, it's a cheek swab. So you take a really long Q-tip, basically, and rub it against the inside of your cheek for a minute, and that collects enough DNA for us to do the analysis. Along with this at-home testing kit, it was important to set up those realistic expectations. So we developed a 4-minute video to talk about the risk, benefits, and limitations for the patient. Once we were able to do that, get patients testing at home, this allowed more access points to pharmacogenomics in the NorthShore system. Incredibly important. We were able to exponentially grow the number of patients being tested any given day at NorthShore while still ensuring they have realistic expectations of what's going to happen with their testing. So figuring out a mechanism around this in your healthcare system could be crucial to the success of a pharmacogenomics program. Once we had this data, we then had to deliver it to the provider. So we're talking about that area in the blue here. So at NorthShore, our data flow is we have many ins, which are represented on the left side. They go to a genomic repository. That genomic repository is where we standardize everything. We link it up with a knowledge base, and then we push it out into a number of different components, including electronic medical record and additional applications such as a pharmacogenomics profile. This approach allows us to meet the many needs of providers without knowing the specific need that they're going to use, because they have a multimodal approach and they can find out which one works best for them. It was important while we were doing this to recognize that there are 2 different levels of clinical interactions with pharmacogenomics, and based on these different levels, we probably needed to develop different delivery mechanisms. So we have the highest risk drug profiles. And so these are the ones where the genetics suggest that getting even 1 or 2 doses of this medication may be detrimental to the patient, and we need to do something immediately. The greatest example I have there is CYP2C19, poor metabolizers in clopidogrel. So in the setting of high-risk acute coronary syndrome, patients may be stented. That stent has the possibility of being closed or rethrombosing because platelets will aggregate to the outside of the stent. You give a patient clopidogrel to prevent that aggregation of the platelets. If the platelets aggregate, closes up the stent, the patient has a heart attack that could ultimately lead to their demise. Want to avoid that. Well, in poor metabolizer patients, they cannot activate clopidogrel as the average patient. And so if you gave a poor metabolizer clopidogrel, it's like giving them a placebo, so increasing their risk of death. So it's definitely something we want to try to avoid, and it's going to be this high-risk profile. And you'll notice at NorthShore, most patients don't have high-risk drug interactions, and so this gives us some flexibility as to how we might want to deliver it to patients or to providers, maybe as an interruptive alert. And it's going to be really important. I'll show you that in a second. When we compare that to medium risk interactions, and so these are ones where knowing the genomics is really can be helpful to the patient and provider. And if you look at these 2 graphs, what you'll notice is the number of cautions per patient in this medium risk is much greater than in the high risk. And so we're talking about 15 to 25 interactions for most patients. And I'll give you an example of what this interaction looks like. So let's say this is OPRM1, poor responders to opioids. And so this says these patients may need higher doses of opioids than the provider might otherwise expect based off the clinical picture. And so if a provider was having to deal with 15 to 25 interruptive alerts per patient, they're going to get burned out really, really quickly. And so based off of these risk profiles, we needed to develop 2 different mechanisms for delivering this information to providers, but still being able to deliver to them in useful ways. And so for those high-risk ones where most patients don't have the profile to trigger anything, we thought interruptive alerts would be a great way of doing that. So you can think about this as active CDS. The system goes out and does something. Here's the clopidogrel example. We let this provider know that this patient is at risk of decreased efficacy. We suggest using an alternative agent, and we set them up to remove the existing order and put on a new order. And this would interrupt their workload to help prevent that catastrophic event from happening. But for those other events, the passive CDS, we've actually created a pharmacogenomics profile, and we use a third-party company, which you can see in the screenshot, called ACTX, to help us deliver this to the medication profile for the patient. And so as the provider is in the portal or in their electronic medical record, they can click on this portal and they'll see an alphabetical list of medications that are a problem for the patient, what the problem is, and then finally the genetics. And so this profile is really interesting because we're meeting the provider where they need to be. We're giving them this data from the drug point of view, that thing they're most familiar with, as opposed to leading out with the genetics, because we found out that genetics, they're really not comfortable with it. And so we wanted to avoid the genetics where we can and get them to that thing that they understand best, the drugs. And so this profile allows us to do this. And patients will have 20 to 30 medications on these profiles. And it does bring up the question of, are there better ways to do this than even this drug profile? We're going to see what that goes in the future. All right, and so the last component that we're going to talk about is going to be education. Education is incredibly important for pharmacogenomics. And what does that education look like? Well, it's the clinical knowledge. It's the educating about why pharmacogenomics is important. What are the drug-gene pairs where pharmacogenomics matters? What does testing interpretation look like? So when they get a poor metabolizer phenotype, what can they extrapolate that to? And then it's also understanding the ethical, legal, and social issues associated with pharmacogenomics, so that any provider that a patient sees at NorthShore is empowered to answer questions and meet the needs of the patient regardless of what's going on. But in addition to that, it's the operational knowledge. It's how to use the tools that we've developed so far, teaching them how to order a pharmacogenomics panel, how to look at the profile, how to respond to alerts, where to find it in the record. And a lot of people forget about this operational knowledge that you have to deliver to providers, because at the end of the day, I could test patients all day long, build all these really cool tools, but if nobody knows where to find them or how to use them, we're never going to derive value for pharmacogenomics. We're never going to derive value from that testing the patient agreed to. All right, as we begin to wrap up, I want to emphasize a few points. So this is the timeline for NorthShore's pharmacogenomics program. So before I arrived at NorthShore in 2014, we have what I like to call the wild, wild west of pharmacogenomics at NorthShore. So this is where clinicians are ordering pharmacogenomics with little to no support from the healthcare system. So we have no idea what was going on in this time, but we have found out through some other patient interactions that there were patients getting pharmacogenomic results that had value, but nobody was acting upon the value for those results. In August 2014, around the time that I joined, we created a pharmacogenomics subcommittee of P&T, and we gave ourselves some oversight infrastructure that began to allow us to tackle small projects moving towards the large implementation that we have today. About 5 months later, we had created interruptive alerts for 3 drug gene pairs. So we started really small. We looked at drug gene pairs where the logic behind them were pretty simple and where the prescribing volume was pretty low, so we could cut our teeth on the low-hanging fruit. And if we got it wrong, it wasn't going to impact that many patients. As I mentioned before, in March of 2015, we created the Pharmacogenomics Clinic. And again, that was because we started to support pharmacogenomics, and as we supported it, more and more people became interested in it. We needed to meet that need of our patients and providers. Very important to understand their need and go out and meet it as you're implementing pharmacogenomics. A year later, we moved to that third party that we talked about earlier to enhance our clinical decision support. And so we went from supporting 3 drug gene pairs in the medical record to doing 9 and 45 different drugs. So exponentially grew what we were supporting with clinical decision support. In October of 2016, we moved to in-house testing, and that quickly led to, in January of 2017, it's hard to imagine, it's over a year ago now, we had the ability for any provider in NorthShore to order that test through the on-demand educational video that patients can watch online, and then the at-home testing. And this has allowed us to test many more patients than we ever could have through the clinic. So we were ordering about 15 tests a month through the clinic, maybe 30 at the most, to just recently in December, we had ordered 160 tests across the healthcare system. So you can see how much we grew. So we started out small and took small projects and built upon them to establish a program that is pervasive across the healthcare system. I'm going to show you a couple of graphs to show you how things have changed over time. Early on, our departments in 2015, who were using us? Well, it was mostly psychiatry. You might ask, well, why was that? Well, one, the best data for the value of pharmacogenomics was available in the psychiatric population. And those were the providers that showed the most interest, so they're the ones we went after first. And then we got more stories based on psychiatry, and so more psychiatrists, it began to resonate with them. But one of the things that we found was patients weren't getting their first antidepressant, their first antipsychotic when they were with the psychiatrist. They actually got those medications before that with their primary care physicians. And so if we look 2 years later, we can see that primary care is actually our greatest utilizers of pharmacogenomics at NorthShore. It gets to that idea that we're pushing preemptive testing. Test the patient before they have the medication. Test them where we can gain the most value for pharmacogenomics. So next steps, we continue to update the system based off new data that is found, and we're looking at the best mechanisms around how do you keep the system up to date. And can we do that leveraging external resources? The more work we can get third parties to use, international consortiums like CPIC, the less work we have to do in our system, the more scalable our approach. We believe that risk score-based interventions becomes incredibly important. We can see that in a number of areas such as prostate cancer or cardiology, and we think some of these risk scores may be able to move into pharmacogenomics if we can predict patients who are likely to get high-risk medications before they have them. And then we believe that the genomics is just the first omics that's going to be important in pharmacotherapy. We think proteomics is going to be important, metabolomics. Already some great emerging evidence of microbiomics becoming incredibly important to drug response. So how can we integrate that into the system we already have? Those are all challenges I'm looking forward to for the next 1 year to 5 to 10 years. So in conclusion, key takeaways for the present for implementing pharmacogenomics: start with small, manageable projects based on your resources. Knowing your resources is incredibly important. Collaboration is key. Implementing pharmacogenomics is more than just the pharmacist. It's the whole healthcare team together, and it includes individuals from a number of disciplines. So collaborate where you can. Additionally, we're not doing anything new with pharmacogenomics. We're just delivering new data. So a number of the processes that you have had already developed in your healthcare system can be used for pharmacogenomics. So identify those and capitalize on all of that work already done. And pharmacogenetics works best if it's integrated into the standard workflows of your institution and of your clinicians. Don't make them come to you. Meet them where they're at and at their needs, so that the valuable work you get done can actually impact patient care. And with that, I will take questions and open the floor. Thank you for your attention today.
Thank you, Dr. Dunnenberger, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many questions as we have time for. So let's get started. Our first question is, what clinical area should be addressed first when implementing pharmacogenomics?
So this is a great question, and it's a question that has many answers. Unfortunately, there's not one main go-to area. I think when you're looking at which area should you start doing pharmacogenomic implementation at, it's understanding who at your institution is supportive of doing pharmacogenomics. So that's both supportive from the administrative standpoint, how much resources do you have available to you, and also from the clinical standpoint, who's really, really interested in doing pharmacogenomics. So if you look at programs such as St. Jude Children's Research Hospital, one of the areas they started with was pediatric ALL. Why? Because that was a huge need at their institution. They were treating a lot of patients with pediatric ALL, and that's where they had expertise. So they went after that one first. It was one they understood the best. If we look at the University of Florida, their cardiologists were really interested in CYP2C19 and clopidogrel and cardiology pharmacogenomics. So they started there first. But if we look at NorthShore, well, we didn't start with pediatric ALL because that population didn't exist for us. We talked to the cardiologists, and they weren't sold on the clinical data available at the time when we first started to implement pharmacogenomics. So we didn't go after that. But instead, our psychiatrists were really interested in understanding how do we use pharmacogenomics. So we went after them first on the clinical implementation side. But because the genetics around depression and psychiatry is complicated, it's multiple genes, we didn't actually systematically support that first. We were supporting that through clinical consultations. And we went after actually the thiopurines and TPMT from the rheumatological standpoint first, because that logic was well established and it was an easy way of building in Epic, our electronic medical record. So we actually took two different approaches, but identifying what resources we had, who was excited, and where we thought we could get some major momentum. And then we began to grow those together so that we began to have overlap between the two of them. So it's really important to understand where are your resources, who is available, who wants to work with you on pharmacogenomics, what patient population do you have that can benefit from this, and pick the one that's right for you. Unfortunately, I don't have the answer for this is the right one to go to every time, but there's probably no one wrong answer here. There are a lot of right answers for you to go after. So thanks for that question.
Our next question is, what is the biggest challenge facing your program today?
So the biggest challenge facing our program today, actually, I'm not going to answer it as one challenge. I'm going to answer it as multiple challenges, because I think on each front, we have different challenges. So one is we've done a lot of work at NorthShore on getting this data to the provider, and we need to better understand how do we get this data to the patient. Patients are probably the best vehicle for disseminating this data across one institution as well as multiple institutions. At NorthShore, we have a number of patients that receive care at multiple health systems. And those other health systems don't have the informatics support that we have at NorthShore, but those patients should still be able to benefit from the pharmacogenomic testing that we have done. And so we have to figure out how do we deliver this data to patients in a way in which they can understand it, and a way in which they can understand it and then educate their other providers about it. So we're trying to figure out what that looks like. It's probably a patient-based portal, a patient-facing portal with a lot of information. Another challenge that we have is as we've developed pharmacogenomics, other genomics have become important. So hereditary cancer genomics, risk for cardiomyopathy, sudden cardiac death. That's becoming an area of interest at our healthcare system. And so we're trying to understand how do we integrate all of that so that clinicians have one genomic place to go, as opposed to having a place to go for this genomic or that genomics or those genomics, because we think if we do that, ultimately they're going to lose interest in genomics. And that brings me to the third challenge that we're facing today. And that's going to be keeping up interest in pharmacogenomics for those providers who have not already adopted pharmacogenomics. So when the tools are new and shiny, it's pretty easy to get people's attention. But you can see it with any implementation, not just pharmacogenomics. Over time, clinicians, a number of them lose interest and stop using the tools that you've built. And so you have to go in and do re-education. In addition to doing that re-education, you have to begin to understand, why did they lose interest? What didn't we do right that first time? And so having a system of evaluating and improving and implementing and evaluating and improving and implementing over time, that quality of improvement system that we've learned so much about through medical errors and other parts of clinical implementation is also true here. So taking your tools and making them better every time to meet the needs of your providers, because if you don't meet the needs of your providers, why are you doing implementation? So those are really three areas that we're challenged with today. And so again, how do we deliver this information to patients? So that's actually another user of our data. And also, how are we going to get providers on board who are not already on board? So thanks for that question as well.
And it looks like we have time for one more question. What pharmacogenetic test do you use in your institution?
Yeah, so this pharmacogenomic test that we use our institution, it's a 20-gene panel, looks at about 120 different variants, and it's a panel that we customized for our own institution, and we run it in our in-house laboratory. And this is a good approach for us, partly because we have the resources in the laboratory space. But when we originally started, we did not use this test. We actually used some third-party testing. It's probably a question that I can answer through a whole other presentation. I'll spare you those details today, but identifying a pharmacogenomic test that meets the needs of your patients and providers is incredibly important. What do I mean by needs? I mean testing the genes and variants that are important for whatever drugs you're going to do the implementation, whatever clinical area that you're going to implement on, as well as testing the variants that are found in the ethnic mix of your population is going to be important as well. Otherwise, you may get some erroneous answers. You can look at warfarin pharmacogenomics to see the importance of testing variants that are seen in the ethnic mix of your population. There are a number of tests out there in the commercial space. Having a thorough way of evaluating them and making sure they're meeting the needs of your health system is going to be incredibly important when you do this. having a systematic approach is incredibly important. There are some papers on the literature that can help you go through this process. It's a really fundamental question you have to answer early on. Again, it's one of those ones where there's multiple answers out there. For NorthShore, the right idea for us was to do it ourselves, but I know for a number of other institutions that's not the right approach. So don't go to it because you think it's the best thing to do unless you have the support to do it. There are a number of great third-party labs out there. All right, thanks for that question as well. And I think that concludes today. So thank you for your attention, and I look forward to seeing your guys' success in the future with implementing pharmacogenomics.
I would like to once again thank Dr. Dunnenberger for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.